Hello. I hope everyone is okay. Today we'll be talking about enzymes. So this is a block of 4 lectures dealing with at the end cellular respiration. Now, cellular respiration is mediated by many different enzymes. So it's a good idea to talk about enzymes to begin with. So these are the learning outcomes. Okay, we'll talk about the different roles and the properties of enzymes. Okay. We'll also look at the diversity of enzyme catalysis. Okay, there are different mechanisms by which enzymes perform their functions, and based on the way they perform their functions, the enzymes can be classified as well. Okay, and then we'll talk about how enzymes work on specific substrates. Now, there's a degree of specificity here. Okay, some enzymes will be extremely specific. Some enzymes will be vaguely specific. Okay, so there's a degree of specificity here. Okay, and then we'll finish off by looking at how enzymes perform their main function, which is to catalyze reactions. Okay, so when we look at the whole genome, okay, we know that DNA codes for proteins. Okay, now the genome has been characterized. Okay, we know roughly what each section of the DNA codes for. Okay, now based on the sequence data that we have from the Genome Project, we can confidently say that about 25% of the total protein, okay, content um, is are enzymes. Okay, in other words, about 25% of the genome. Okay, so if we look at this green sector here, about 25%, okay, um, of the genome codes for proteins which are specifically enzymes. Okay, there are other proteins as well, but the ones which code specifically for enzymes amount to 25%. Okay, so you can see when you compare that to say something like immunity here. Okay, we know how important immunity is, but look how much of the DNA codes for immunity specific proteins. Very small compared to, say, enzymes. So we can say that confidently that enzymes are extremely important. Okay. Now, hopefully everybody knows what enzymes are. Okay. Enzymes are mostly protein molecules whose job it is to speed up reactions, or in other words, to catalyze reactions. Okay. Now, one way we can picture this quite easily, okay, is by looking at this reaction here. So this reaction here describes the formation of carbonic acid from carbon dioxide and water. So carbon dioxide reacts with water to form carbonic acid. It's something that we've talked about many times before. Okay, but this is an important reaction when it comes to things like putting fizz into drinks. Okay, the way it works is that we're pumping carbon dioxide into, say, something like water. Okay, at high pressure, that carbon dioxide reacts with water. It dissolves in water, and what you get is the formation of carbonic acid. Okay, now you can see over here this is a reversible reaction, so it goes forward to form carbonic acid, and carbonic acid can dissociate to go the other way to form its substituent molecules, carbon dioxide and water. So we put carbon dioxide in, and carbon dioxide is a gas. Okay, but then when carbonic acid dissociates to form the gas again, this is what we see as bubbles. This is the fizz. So carbon dioxide is the bubbles that make up fizz. Okay. Now, the interesting thing that you see when you pour when you pour a glass of fizzy water into a glass, okay, you'll see lots of bubbles being produced, which means that this reaction here, carbonic acid to carbon dioxide, is really fast. Okay. But then after a while, what happens is that it settles down. So after a while, only a few bubbles will develop to rise to the top. Okay, so you see that with the odd bubble being produced at the bottom, on the sides, the glass rising to the top. So in other words, in the beginning, when you poured the water, fizzy water into the glass, this reaction was really fast. Okay, but after a while, it settles down, and this reaction starts to become slower. Okay, now if you take a sip of this water, fizzy water, okay, and you take a sip, what you notice is that there's an explosion of bubbles of fizz in your mouth. Okay, now why is that the case? Okay, so well, first there's a physical reason. Okay, you're putting it into your mouth, you're exaggerating, you're speeding up this reaction anyway, just by disturbing this carbonic acid. Okay, but another thing, the reason why you create so much fizz is that your mouth, okay, obviously made up of cells, express this particular enzyme, carbonic anhydrase. Again, it's an enzyme which we've talked about before. Okay, it's important on red blood cells. Okay, uh, but it's an enzyme which essentially speeds up this process. Okay, so carbon dioxide reacting with water to form carbonic acid. So it speeds up that process, but the enzyme can also speed up the reverse process as well. Now, in the mouth, it speeds up the reverse process. There's lots of it. Okay, there's lots of carbonic acid dissolved in the water, and so the carbonic anhydrase, okay, speeds up the reverse reaction. Okay, and so in other words, you speed up the reaction, you speed up the rate of producing carbon dioxide. Okay, which exists as which obviously is is a gas. Okay, so it comes up as bubbles. Okay, so as soon as you put some fizzy drink into your mouth, okay, the carbonic anhydrase speeds up the reaction going from carbonic acid to carbon dioxide. Okay, so that's kind of a straightforward example of what enzymes do or how enzymes work. We know how important enzymes are. We said before that without enzymes, there is no life. Okay, because if you take this reaction here, which we rely on, so we take in glucose and we take the energy that's locked away in glucose, okay, to form ATP. So this is the cellular respiration equation. Okay, and the main purpose of burning, or I wouldn't say burning, but breaking down glucose to form is to produce ATP. We need oxygen, okay, in the reaction for this to happen. Okay, so sugar in the presence of oxygen, in the presence of a multitude of enzymes. So this arrow really represents a multitude of enzymes, which after all these different reactions gives you ATP. It also gives you carbon dioxide and water. Okay, but the main purpose of this reaction is to produce ATP in our bodies. Now, when you take sugar, okay, so if we take sugar and leave it on the shelf, there's lots of oxygen in the air, okay, but we don't really see any carbon dioxide produced. Ask yourself the question that why isn't this the case? Because if I take a step back now and just let you know, I mean, you probably already know this, is that enzymes don't invent reactions. So this reaction isn't invented. This reaction is going to happen uh, whether you like it or not. What enzymes do is just speed up existing reactions. So this is a reaction that would happen in the absence of enzymes. So this is the so we take sugar in the form of glucose. There's oxygen in the air, okay, uh, and we produce CO2. But if you leave it, if you leave some sugar on the shelf, let it react with O2, we don't see any carbon dioxide. Why is that the case? Well, when I say there's no carbon dioxide produced, there's no perceptible levels of carbon dioxide produced. In other words, the reaction here is so slow that we can't really measure the carbon dioxide coming off from the natural reaction of oxygen with sugar. Okay, so this reaction is extremely slow. Okay, and if we relied on the speed of the natural reaction, we will nowhere near produce enough ATP, okay, for our cells to survive, for our tissues to survive, and for our organs to be viable, and then for the organism to survive, to live. Okay, and this is why we need enzymes, so that this reaction here speeds up sufficiently, adequately, that we can produce the amount of ATP that we need at any given moment. Okay, so if we left this sugar on the shelf and let it and left it for years and years, and I'm talking, when I say years and years, I'm saying, 50 years, 100 is maybe even longer than that. Okay, when you come back after 100 years, maybe the sugar will have disappeared. It will have oxidized from the oxygen in the air, okay, to give you carbon dioxide. But that's how long it would take. Okay, hundreds, maybe 100 more, maybe more than 100 years. Okay, in our bodies, this process is sped up so quickly that it can take microseconds. Okay, so this process is sped up, it is catalyzed, okay, by enzymes. But remember, it's not one enzyme, it is a whole series of enzymes. And this is something we'll talk about when we talk about cellular respiration. So just to sort of reiterate that, here's a glucose molecule. Look how complex it is compared to the product carbon dioxide. Okay, carbon double bonded to one oxygen and double bonded to another oxygen. Compare that to the original glucose molecule from which this carbon dioxide came from. Look how complex it is. Okay, so what we're basically doing is we're breaking the bonds, okay, through oxidation, through the presence of oxygen. Okay, it's much more complex than that. Okay, we're basically removing electrons, high energy electrons, which bind carbon and hydrogen together. That's what we're basically doing. We're removing the electrons, the high energy electrons in these carbon hydrogen bonds. Okay, and as we're doing it, and we also do this, our bodies, our cells do this in a very stepwise fashion. Okay, not all of it at once. We don't break all these carbon hydrogen bonds at once. We break them in a stepwise manner, and as we break them break them up in a stepwise manner, we're sort of yielding, releasing the high energy electrons, okay, which then are picked up by the oxygen, okay, in cellular respiration, okay. And those high energy electrons that we've extracted from these is used to power the production of ATP. Okay. So like I said, like it says over here, this is a very complex process. Okay. We write this reaction, but it's mediated by many different enzymes. So it's a complex process that requires many enzymes for this reaction to be completed. Okay. So so now that we know that enzymes speed up reactions, okay, how fast are these reactions actually? Okay. And you can see from these numbers here that they are extremely fast. Okay. Now, if we take this reaction that we've seen before, carbon dioxide reacting with water to form carbonic acid, okay, this this reaction happens at 0.01 molecules per second. Okay. In other words, you would need, so if you take this decimal place 10, 100, so you would need 100 seconds to produce one molecule of carbonic acid. Okay, compare that in the presence, compare the reaction in the presence of carbonic anhydrase, and now you're producing 100,000 molecules per second. Okay, so this would be, so you would, in the absence of the enzyme, you would need 100 seconds just to produce one molecule. Okay, And in the presence of enzyme, just in 1 second, you're producing 100,000 molecules. Okay. Now, that is a 10 to the power 7-fold increase in the production of carbonic acid. Okay. So it's a 10 to the 7 of 10 to the power 7-fold increase. Now, that isn't even one of the fastest enzymes. Okay. There are other enzymes which are much faster. Look at ureas. Okay. An enzyme that's responsible for breaking down urea. Urea is produced as we break down proteins. Okay, and so this enzyme is much faster than carbonic anhydrase. It's not quite as straightforward as that. Okay, as you'll see in a later slide or in a later lecture. Okay, but you can see how fast. So the point of this slide is really just to demonstrate how fast enzymes are, but also note that enzymes don't invent reactions. They just speed up existing reactions. So again, this reaction will happen. Urea will break down by itself. Okay, and it will be oxidized in the presence of oxygen. But ureas can speed up the process by 10 to the power 14-fold. Another feature about enzymes is they perform these reactions, these extremely quick reactions, but they perform these reactions in such mild conditions. Okay, so look at the temperature. Enzymes are made to be active, especially, I mean, when we look at our own mammalian bodies. Okay, we maintain our temperature at 37 degrees Celsius. Our physiological pH is around 7.4, 7.35 to 7.45. Okay, and the pressure in our bodies is normal atmospheric pressure. Okay, normal atmospheric pressure. So these are the conditions in which our enzymes perform these extremely quick reactions. Now compare that to industrial catalysts. Okay, remember, enzymes are just catalysts, biological catalysts, but industrial catalysts. Okay, look at the conditions they need uh, for the reactions to be sped up, to be catalyzed. Okay, if we're looking at, say, something like the industrial manufacture of ammonia, so in making fertilizer. Okay, this requires metals. So the major component are metals in these industrial um, reactions. Okay, because metals act as electron donors, but also electron acceptors as well in some cases. Okay, but you have to heat these metals up to this types of temperatures, 477 degrees Celsius in the manufacture of ammonia. You also have to expose the reaction to such high pressure. Okay, so one bar of atmospheric pressure. This is 200 bars. Okay, so it's 200 times the pressure that our enzymes work on. Okay, that's like diving 2,000 meters underwater, and that 2,000 meters of water pressing down on you. Okay, so obviously we would never survive that. Okay, so these are the kinds of uh, conditions you would need. Okay, if you take something like a catalytic converter, okay, which removes harmful nitrogen oxides and carbon monoxide, okay, as you burn fossil fuels, these these are the sort of substances which are produced, which are toxic. Okay, toxic. They're doing lots of damage in the environment. People breathe it in and suffer all kinds of consequences. Okay, increase in asthma could be linked to the increases in nitrogen oxides, nitrous oxides, and carbon monoxide in the atmosphere. Okay, so by law now, obviously cars have to have catalytic converters, and we're basically uh, speeding up the reaction of the oxidation and reduction of these substances. Okay, so okay, so it's reduction to remove oxygen from nitrogen oxides, nitrous oxides. Okay, to produce harmless nitrogen. Okay, but 70 odd percent of our air is nitrogen. Okay, so we're reducing nitrous oxide compounds, so removing the oxygen to produce harmless nitrogen, and oxidation of carbon monoxide to produce carbon dioxide. Carbon dioxide is a greenhouse gas, but carbon monoxide is a killer. Okay, people breathe it in, and it destroys cellular respiration. Okay, so but anyway, the thing is, back to the point. For this to work, for the catalytic converter to produce to do these reactions, okay, um, the catalytic converter needs to reach a temperature of up to 300 degrees Celsius. Okay, so that's so if your converter doesn't reach these temperatures, okay, the rate at which nitrous oxide is reduced and the rate at which carbon monoxide is oxidized to produce carbon dioxide, okay, doesn't happen really quickly. So in other words, as you be driving along, your car would be your catalytic converter wouldn't be as efficient, but still be producing dangerous nitrous oxides and dangerous carbon monoxide. It's only when you've driven for a while, engine becomes hot, and the catalytic converter becomes hot as a result of that. That's when you get good catalysis of this reaction. Okay, so back to the point. Okay, enzymes perform catalysis under very mild conditions. Although we as human beings have enzymes which work in mild conditions, okay, 37 degrees Celsius and around about neutral pH, 7.35 to 7.45, okay, enzymes are very versatile, okay, and there's all kinds of organisms which grow in extreme conditions. Okay, so underwater volcanic vents, okay, where the temperatures are really high, above 90 degrees Celsius, and you can see from the colors from these volcanic vents that there's so life is flourishing. So we have microorganisms exposed to such high temperatures, thriving in these conditions. Okay, in volcanic lakes, okay, volcanoes release lots of sulfur gas. So this yellow stuff here is all sulfur. Sulfur reacts with water to produce sulfuric acid, and so the pH is extremely acidic. Okay, we can have the opposite as well in soda lakes, where the pH can be very alkaline. Okay, but you can see from these colors, so the sulfur represents sulfur, so the yellows represent sulfur, but they also represent organisms which are thriving in these extreme conditions. Okay, so enzymes don't necessarily have to work in mild conditions; they can be adapted to work in extreme conditions as well. Okay, so if you took these microorganisms and put them in mild conditions, they won't thrive as much as well as they do in these extreme conditions. Okay, so so in other words, these extremophile microorganisms have evolved to flourish at these extreme conditions. So their enzymes, they've adapted, evolved over many thousands of years to you know to benefit from these extreme conditions. In a way, that's not entirely true. These were the original organisms because the original Earth was very toxic to life. Okay, in the early Earth, before there were human beings, before there were mammals and more complex organisms, we had microorganisms which lived in the extreme conditions that Earth posed. Okay, so these were the kind of the original organisms. But the fact remains that their enzymes, okay, have uh, have been adapted to work in these extreme conditions. Okay, now we can take advantage of that. Okay, we can take advantage of that and use those enzymes to our benefit. Now, everyone's been talking about coronavirus, okay, and the fact that people are being tested and traced. Now, the gold standard for detecting coronavirus, somebody has coronavirus or not, is PCR. PCR, polymerase chain reaction. Okay. Now, the way the polymerase chain reaction works is that you take a sample from a person. Okay. The sample, if they have coronavirus, okay, they also have coronavirus RNA. Okay. That RNA initially it's converted to DNA. Okay, and so if there's no coronavirus RNA present, there'll be no the PCR wouldn't work basically. Okay, although the PCR would work, but it wouldn't produce a product. If there was coronavirus RNA, we would be able to detect that in the PCR reaction, that a product would be formed. Now, the way this PCR works is that we have to heat the sample, upwards of 70 odd degrees, or even higher than that, to melt DNA. So, if you remember from the lecture we had on DNA, DNA exists as a double-stranded molecule. Okay, and if you want to replicate DNA, you have to melt the two strands. So you melt the two strands, and you can perform through a DNA polymerase reaction. You can make more DNA. You can make copies of that DNA strand. Okay. Now, in order to melt the DNA, okay, in our bodies, in our sort of mild conditions, we can do this by having special enzymes which unravel the DNA. So you don't need to expose the DNA in our bodies to high temperatures. We have special enzymes which can unravel the DNA. Okay, but in a polymerase chain reaction, okay, we can do this more simply. We don't need the other enzymes to unravel the DNA, because that would make the whole process very slow. So what we do is we melt DNA, okay, just with heat. So we can expose the DNA to high temperatures, up to 80 degrees Celsius, even 95 degrees Celsius, okay, to melt the DNA, so that the DNA polymerase can work. But the DNA polymerase we have in our bodies, okay, if we look at this graph here, our DNA polymerase works efficiently at 37 degrees Celsius. Okay, oops, it is, sorry. Works efficiently at 37 degrees Celsius. Now, either side of 37 degrees Celsius, let's say 40 degrees Celsius, look at the rate of catalysis here. It's dropped dramatically. Or if we drop it under 30 degrees Celsius, it drops dramatically. Okay. So in other words, we could never use our DNA polymerase in the polymerase chain reaction, because at such high temperatures, 95 degrees Celsius, the polymerase would denature and not work. Okay. And this is why we use the DNA polymerase from these extremophiles, which have adapted their DNA polymerase to work at high temperatures. Okay, so look at this. This is the DNA polymerase from one of the extremophiles which work at high temperatures, and you can see that the high temperature, so it maintains its so it works best at around 75 degrees Celsius. You can adapt this for them to work even at high temperatures as well. Okay, but even if it reaches high temperatures, it doesn't get denatured. It falls back into position. So you can expose this enzyme, this DNA polymerase, to even higher temperatures. It won't work as well. Okay, but then when you bring the temperature, the high temperature is really there to melt the DNA. When you bring the temperature back down to 75 degrees Celsius, so in the polymerase chain reaction, that's what you do. Okay, the cycle of PCR. You go from a period where you perform the DNA polymerase reaction, and there's another phase where you melt the DNA. So that's when you go to really high temperatures, and then you bring the temperature back down again, so the DNA polymerase can work much better. Okay, so these sort of enzymes from these extremophiles have adapted, so they work in these these extreme conditions. But the enzymes which we have been are made so that they work at peak physiological conditions. So normal 37 degrees Celsius and normal sort of around about neutral pH 7.35 to 7.45. Okay, so let's just summarize that a little bit. So general properties of enzymes. So obviously we know that they're much faster. Okay, so much higher reaction rates, 10 to the power 6, 10 to the power 14, fall faster than the non-catalyzed reaction. The conditions in which these enzymes work are usually mild in the context of human beings. Okay, normal sort of mild temperatures, 37 degrees Celsius, normal atmospheric pressure, and close to neutral pH. Another feature about enzymes is that they have uh, reaction specificity, okay, compared to chemical catalysis. So before, we talked about some of the industrial catalysts, okay. Now, if you introduced a contaminant in the mix, okay, you would not just produce your product, the product that you want, but you would also produce contaminating products as well, okay, because the reaction isn't specific. As soon as you expose high temperature and high pressures to anything really, okay, they can react non-specifically and produce all kinds of products, okay. But enzymes, you can put anything in the mix, to a certain degree, but you would only get, depending on the enzyme that is, a very specific product, okay. And generally speaking, I mean, we can generalize this to a certain extent that one enzyme recognizes one substrate and produces a specific product. Okay, we can. There's much more nuance to that than this statement implies. Okay, enzymes uh, can have a diversity of substrates, which we'll come on to in a few minutes. Okay. Now, in terms of regulation, okay, we can turn down the temperature, we can turn down the pressure in these industrial catalysis. So there's these industrial catalytic processes. Okay, but when it comes to our enzymes, there's a much more sorry, there's a much more variety in ways which we can actually regulate uh, this catalysis. Okay, so the capacity for regulation is much more varied. Okay, now we have ways by which we can regulate enzymes speed of catalysis, if you like. We can also regulate whether we can whether to switch them on or switch them off. So there's different ways in which by which we can regulate enzyme activity. Okay. Uh, One common way is through allosteric. So certain molecules are produced in the cell, okay, which can bind to the enzyme. They bind sometimes through covalently, okay, and that by doing that, it changes the shape of the enzyme a little bit, and it affects their ability to perform catalysis. Okay, so a common way is through a negative feedback loop. So the actual product that the enzyme makes, if there's sufficient amounts of it, it can then bind back, okay, and inhibit the enzyme so that it tells the enzyme to stop working or to reduce its catalysis. Okay. Another way of regulating enzyme activity is just by simply producing less of it. Okay. So if the enzyme is producing lots of product, that product can not only in some cases bind to the enzyme and inhibit it, but it can also bind to other factors such as the DNA that codes for the enzyme. So it binds to the DNA and switches off the production of more enzyme. Okay, so that way we can actually uh, change the quantity of enzyme in the cell. Okay. Now let's talk about enzymes and their classification. Okay. Now enzymes end with the ending or the suffix ase. Okay, so if you come across these words protease or lipase, okay, because they have ase at the end, ase at the end, it denotes that this particular molecule is an enzyme. Okay, protease which breaks down proteins, lipase which breaks down lipids, so on and so forth. Okay, enzymes are named according to their substrates. Okay, so like proteins, proteases that break down proteins, uh, and lipases which break down lipids. Okay, so they are named according to their substrates, but they're also named or classified by the type of reaction they perform as well, or they catalyze. Okay, now the story of classification is quite a long one, but suffice to say, okay, new enzymes are being uncovered every day. Okay, there are many people around the world working on enzymes or working on all kinds of things, and in the process, they uncover another enzyme. Okay, which is a novel thing, and the more knowledge we have about the different enzymes that exist in the world, the better we are prepared for. Okay, so when a scientific group uncover an enzyme, they'll perform some tests, and then they'll go about naming the enzymes. Okay, now luckily we have a system. We have a system by which we can classify enzymes. Okay, and this classification system was invented by this body here, the International Union of Biochemistry and Molecular Biology. Okay, IUBMB. Okay, so they came up with a classification system by which any member from any scientific group from all around the world can use to classify enzymes. Okay, so the rationale, okay, is very clear in terms of naming enzymes. And before this rationale was invented, people were they were uncovering an enzyme in one part of the world, obviously spoke a different language, and another group uncovered the same enzyme, but they spoke a different language. Okay, and they performed different tests, and so they came up with a different name for the same enzyme. And so one group published their paper, another group published their paper, and what they didn't realize was that the enzyme they were talking about were the same enzyme. Okay, so so because of that, there was a need to produce a rationale, a mechanism by which we can classify enzymes. Okay, and and they came up with this rationale here. Okay, in terms of the type of reaction they catalyze. Okay, and enzymes are also given, once they're classified, they're given an EC number. Okay, an enzyme commission number. Now there are 6 different types of chemical reactions which all enzymes fall under. Okay, and it's these here. Okay, so we have enzymes which perform redox reactions. Okay, oxidoreductases. Okay, so we also have enzymes which transfer molecules. Okay, we have enzymes which break molecules by the use of water. Okay, uh, we have also other enzymes which, similar to hydrolases, okay, also break down uh, other molecules, but not necessarily with the addition of water. Okay, we have isomerases. Okay, isomerase, so carry out converting molecules from the left hand to the right hand form. Okay, and ligases, which basically catalyze joining up of molecules. Okay, now oxidoreductases. Okay, so if you we're going to talk about dehydrogenases in another lecture when we talk about cellular respiration. So some of the enzymes which are responsible for breaking down glucose to produce ATP are dehydrogenases. Okay, so they perform redox reactions. Okay, transferases. So I know there's an example here, but if you look at this example here, so adding amino acids to a growing peptide is done by a peptidyl transferase. Okay, so it's basically this enzyme here is bringing in amino acids, okay, and adding them onto a growing chain to produce the mature polypeptide. Okay, hydrolases, so enzymes which break down things. So, for example, proteases break down proteins. They break down the peptide bonds. Okay, how do they do this? Well, they do it by introducing water, a hydrolytic reaction, to break down these peptide bonds. Okay, liases. Okay, these are a bit like hydrolases, but we don't use water. Not We don't use water directly to break these bonds. Okay, we definitely need water in hydrolytic reactions, but not necessarily in lyases. Okay, so this example I've shown here, so there's this molecule here, AB, which is linked to XY. Okay, and you break it down, so you get the production of AB and XY. But not only that, you've also introduced a double bond. So before there was a single bond, and now you've introduced a double bond. So this is another one of the features of lyases. Okay, that you introduce, but not only introduce, lyases can also do the reverse and go from a double bond to a single bond. Okay, so that's one of the features of lyases. Isomerases, like I said, going from converting a molecule from the left-handed version to the right-handed form, and ligases to join things up. Okay, so things like if we want to join two strands of DNA, okay, we use a ligase. Okay, now when I say strands, you've got to be careful here. You've got one strand which hybridizes with another strand. Okay, those two strands joined together by the hydrogen bonds between the nucleotides. Okay, this I'm talking about the phosphate backbone here. So if you got on one strand, you got one strand of DNA, and you got another strand, and you want to join it not by hybridizing it, but by joining the phosphate backbone bit to form a longer one strand of DNA. Okay, we use DNA ligase for this. Okay. Okay, so this is just to illustrate a bit of repetition here, just to illustrate the different types of reactions in a bit more detail. Here, okay, so I'll skip this. Just have a look at these reactions, see if they make sense to you. If you find that they don't make any sense, just give me a shout, send me an email. Okay, now the lyases, okay, which have an EC number of 4, and maybe I should go back here. So if I go back here, every type of enzyme will have an EC number. So an EC number of 1 illustrates or indicates that it's an oxidoreductase. Lyases have an EC number of 4. Okay, and the enzyme carbonic anhydrase is a lyase. Okay, remember one of the features of this was that it introduces or breaks up a double bond. Okay, now this is the reaction. This is quite complicated. Okay, but what this represents here is the active site of the enzyme of the carbonic anhydrase. Okay, so there's these histidines that make up the catalytic site. Okay, and you also have a zinc ion as well. So this whole area is really positively charged. You've got the water molecule as well attached to it, and because it's very positively charged, it snatches one of the electrons, or it snatches the electrons from the oxygen. Okay, as it does that, one of the hydrogens is released. And what you're left with is a very electronegative hydroxide group here. Okay, and this is when carbon dioxide enters the reaction. So carbon dioxide enters the reaction here, and because this is very electronegative, okay, you get a nucleophilic attack on this carbon dioxide. Okay, uh, this nucleophilic attack produces this substance here, or this molecule. Okay, which is this here, HCO3. This is bicarbonate. Okay, one H, one carbon, CO3, 3 oxygens. Okay, this molecule here, HCO3. Okay, and this bicarbonate is released into the plasma, and so the CO2 that originally existed like this becomes bicarbonate, which is released as bicarbonate into the plasma. Okay, so CO2 doesn't bind to hemoglobin; it becomes bicarbonate. Okay, and then it's taken to the lungs, and then get the reverse reaction. The bicarbonate becomes carbon dioxide, and we breathe it out. Okay, but the fact is here, coming back to the point, is that what we've done here, we've removed a double bond from carbon dioxide. Okay, so carbon dioxide here is double bonded, to one oxygen and double bonded to another oxygen, and now it's got a single bond here. So we've removed a double bond. Okay, and it's also breaking up of a molecule. So this will then dissociate, so it breaks up. HCO3 is released. So we've fulfilled the mechanism of a lyase, introducing or breaking of double bonds, and also we're breaking up the molecules by the release of this HCO3 molecule, the bicarbonate. Okay, so don't worry if you don't get this. We're not going to test you on the process of of catalysis here. Okay, but it's just to demonstrate how lyases work and the defining feature of lyases, in so much that it's that a double bond is introduced or broken up. Okay, and that lyases break molecules. Okay, right then. Okay, so enzymes can be classified. So, for example, if we take our enzymes, carbonic anhydrase, a human carbonic anhydrase, okay, we know that it's a lyase. So its first number is a 4. Okay, all lyases are 4, but then there are many different types of lyases. Okay, so we can specify in much more detail. So our specific carbonic anhydrase is a carbon oxygen lyase. Okay, and so we give that a number 2. There's number 1, 3, 4, 5, etc. For other types of atoms involved. Okay, uh, water is involved at some point. Okay, you'll see that if you go back to the previous slide, water did come in, but it wasn't responsible for breaking it up. Okay, so it is water still involved, so it is a hydro and it is a lyase. Okay, and so this is the EC number given to human beings carbonic anhydrase. It's a lyase, it's a carbon oxygen lyase, it's a carbon oxygen hydrolyase. Okay, and it's the specific carbon oxygen lyase from human beings. Okay, so this is the number. Now, this is important because when you're reading papers, and you'll find yourself reading papers about enzymes, okay, or the discovery of an enzyme. And what you'll find is that they'll say this protein or this enzyme has this EC number. Okay, now this might throw you off if you don't know what EC numbers are. It'll throw you off a little bit. Okay, and so now that you know about EC numbers, you know what they're referring to. Okay, so it shouldn't throw you off now. Now let's talk about specificity of enzymes. Okay, specificity of enzymes. Enzymes, we say, are quite specific in terms of the product they produce. Now, they produce a specific product because they bind specific substrates. Okay, so there's enzymes that are said to be specific for substrates. Now, they're specific for substrates because the substrate has a complementary structure to the active site of the enzyme. Okay, so broadly speaking, the substrate's three-dimensional structure fits in to the three-dimensional structure of the active site of the enzyme. Okay, if they both come together, then the enzyme can perform its catalytic reaction. Okay, so that's why you can have all kinds of molecules in the mix. Other molecules will simply not fit in there, and only the substrate that's specific for the enzyme will fit in. Okay, and only when the substrate binds to the active site will we get a product formed. Okay, now there's a degree of specificity here. Okay, this seems to indicate that there's absolute specificity. Okay, but as you'll find later on, that is not the case exactly. Okay, now we say that enzymes show in terms of the hypothesis, in terms of how they work, how the substrate binds to the to the active site, we say that's analogous to a lock and key process. Okay, so in other words, to open up a specific lock, you would need a specific key. The shape of this bit here of the key fits in quite nicely is complementary to the structure in the lock, this key fits in, and then you can turn the lock. If it doesn't quite fit in, you can't turn the lock. Okay, so enzymes are similar. Okay, so the enzymes structure here, you can see, fits in quite nicely to the active side of the enzyme. Okay, the substrate binds, and the enzyme can perform its uh, catalytic reactions. Okay. Now, in order for substrates to bind to the active side, okay, there has to be uh, some kind of attraction. Okay, uh, what and in a way, there is a little bit of attraction between the substrate and the enzyme. Okay, and that's based on, it's mostly based on, so the vast majority of the reason why a substrate binds to the active side is the three-dimensional structure, but there are also opposite charges as well. Okay, so you can see over here in the enzyme here, there's a positive charge here, and there's a negative charge on the substrate. Okay, what these charges are not so much, they're not so much all about attracting the substrate, okay, but rather holding the substrate in position. Okay, so once the substrate binds through its complementary three-dimensional structure, it remains bound there because of the opposite charges. Not necessarily opposite charges, but just complementary charges. Okay, because the enzyme, so the substrate can go in, but then it can quite easily fall back out. Okay, so there needs to be something there in there that holds it into position, and it's usually these electrostatic interactions. Not only that, but also hydrophobic interactions as well. Okay, so there might be bits over here which are hydrophobic. Okay, and so there'll be hydrophobic parts on the complementary part of the active side. Okay, so you get these interactions, electrostatic interactions and hydrophobic interactions that hold the substrate in the active side. Okay, and once it's held into position, that's when the enzyme can actually perform its catalytic reaction. Okay, so if I just move one slide. Now, if I go back, this seems to suggest, this lock and key method, that if you have uh, if you don't have the substrate that fits in exactly to the active side, then it won't work. What we know in reality is that that's not quite true. Okay, the biggest feature that drives interaction between substrate and enzyme is the three-dimensional structure, the complementary three-dimensional structure of the substrate and the enzyme's active side. Okay, but the active sites, um, so the active site structure is not 100% complementary to the substrate. Okay, so because of that, what can happen is that you can have a range of different substrates which show very similar structures that can all bind to the active side. Okay, and that explains why certain enzymes actually have many substrates, but all those substrates are somewhat very similar. They're not identical, but somewhat similar. So to explain that, that lock and key hypothesis has been adapted to incorporate some flexibility. Okay, so we've adapted that lock and key hypothesis to this induced fit hypothesis. Okay, now what this basically means is that once the substrate binds, it binds very tentatively. Okay, so it binds very tentatively, um, and upon that tentative binding, that enzyme's active site gets triggered to change its shape a little bit so that it fits better, so that the substrate fits better to the active site. Okay, so in other words, the enzyme's active site is induced to fit the substrate better. Okay. So the substrate binds tentatively, it changes the conformation of the enzyme a little bit, and that conformation change enables a tighter interaction between the enzyme and the substrate. Okay, and that's what you need. You need a very tight interaction between the enzyme and the substrate for catalysis to work properly. Okay, so that's the induced fit hypothesis. Okay, so to help illustrate that the induced fit hypothesis, let's look at a real life example. Okay, now this is an enzyme, hexokinase. It's an enzyme which we'll talk about um, in a later lecture on cellular respiration. So this is hexokinase, an enzyme which binds uh, any sugar molecule which has 6 carbons. Okay, hexokinase. Um, so this is hexokinase without the substrate bound, without the sugar bound. So take note of the shape. Now this is hexokinase with the substrate bound, with the sugar bound. You can see here's the pocket which makes up the catalytic site. Look at the so this bit's exposed to the this is the active site, but then look at this bit here. It closes up as well. Okay, so in other words, the sort of overall active site of the enzyme has changed. Okay, not this great big blob here, but it's just this active site. It's changed its shape so that to allow for tighter binding between the substrate and the enzyme. Okay, so we know that this induced fit principle works. Okay, um, but there are some enzymes which don't change their shape. Okay, so the induced fit principle doesn't work with those enzymes. Okay, but there are some enzymes whose shape requires whose substrates are very specific, that only a specific substrate will bind to the active site, and in those cases, it's more the lock and key hypothesis. Okay, so there's 4 types of specificity. Okay, there's what we call absolute specificity. Okay, so the enzyme is specific for one substrate and full stop. Okay, so urease, for example, acts only on urea. Okay, glucose oxidase only works on glucose, as opposed to say hexokinase. Okay, hexokinase works on not only on glucose, but it can also work on fructose as well. Fructose is another 6 carbon sugar molecule. Okay, but glucose oxidase only works on glucose, for example. Okay, so these types of enzymes show absolute specificity. Okay, and then we have relative specificity. Okay, and this is when the enzyme shows specificity to a range of related substrates. An example of this would be pancreatic lipase. Okay, so your pancreas, sorry, let's just go back. The pancreas releases all kinds of enzymes, proteases and lipases. Okay, um, to help with digestion. Okay, so this pancreatic lipase, okay, shows relative specificity because it can break up any ester bond. Okay, remember triglycerides are joined together by ester bonds. Okay, so these pancreatic lipases can break up any ester bonds. So it doesn't really matter which fats are exposed to this pancreatic lipase. If those fats do have ester bonds, then this lipase will break those fats down. Okay, and then we have group specificity. Okay, so there's um, less, I guess, very high specificity. As you go down, less and less specificity. Okay, so group specificity is less than relative specificity. Okay, and the example that I've given here is trypsin. Okay, now trypsin breaks down proteins, but it breaks down proteins it breaks down any peptide bond, provided it has an arginine and a lysine close together. Okay, so if you look at the active site of trypsin, okay, it accommodates arginine and lysine close together. So if you have a long massive protein, okay, and and there's an arginine and lysine at certain parts, that protein would only be broken up at those certain locations where there's an arginine and lysine. So it doesn't really matter which protein you give to the trypsin, provided it has an arginine and lysine close together, either next to each other or separated by an amino acid or a couple of amino acids. Nevertheless, that still fits into the active site of trypsin. Okay, and so trypsin can break that down, can break those proteins down. Okay, and then there's and then there's stereospecificity. So specific for the left-handed version or the right-handed version of the substrates. Okay, so that's quite broad. That's even much more broad in terms of specificity. Okay, so those are the types of specificities which enzymes demonstrate. Okay, I'm going to now just skip through these questions. Okay. Um, Now let's look at how enzymes work mechanistically. Okay, so remember what I said before, that enzymes don't invent reactions. They don't. They just speed up existing reactions. Okay, so enzymes increase the rate of reaction, not whether a reaction takes place or not. Okay, so I mean, we used that example before about carbon dioxide reacting with water to produce carbonic acid. Okay, this reaction happens in nature by itself. Okay, we see it all the time in the air. So lots of carbon dioxide in the air. It reacts with water vapor. It gets into the clouds and acid, and then rain falls as slightly acidic because it has carbonic acid. So that reaction happens nevertheless. Okay, but enzymes speed this reaction up by many by up to 10 million fold. Okay. Now, whether a reaction happens or not depends on what we call the free energy change or the available energy change. Okay. Now, to help, and what I mean by this, we need to look at the second law of thermodynamics, entropy. Okay. So let's just explore that a little bit. Okay. So second law of thermodynamics, entropy. Okay. Now, entropy is a measure of randomness. Okay. Now, the whole universe, okay, is governed by the laws of physics. Okay. And and the universe prefers randomness because randomness, okay, allows for energy not to be concentrated in any one particular area. Okay. Now, if we look at something like this, so look at these molecules. There could be atoms or molecules close together. Okay. Now, there are forces in there. There's lots of energy trapped in there. Okay, so there's lots of energy concentrated in here, and that's important because that energy holds the structure together. Okay, now the universe doesn't like this. Okay, it favors this to be more like this and even more dispersed. Okay, now this sample here is said to have low entropy. Okay, and it's a bit in terms of energy, it's a bit opposite. Low entropy means it has lots of energy within the system, whereas high entropy means it has low energy within the system. Okay, so in order to maintain this structure, energy is required. Okay, in order to hold things together, so if something is structurally ordered, we need energy in it. That's why a glucose molecule, lots of carbon hydrogen bonds, to hold that together requires lots of energy, and that's the energy which we extract for in cellular respiration. Okay, and so that molecule will automatically break down. It'll take forever, but it will break down. Okay, because the universe dictates that to happen. Okay, now okay, so let's look at this free energy then. So the energy held within the structured system, okay, gets converted, okay, to another form. Remember, energy cannot be created nor destroyed. It just goes from one form to another. So the energy in here, locked away in here, gets converted. Okay, so this wiggly arrow it illustrates and the formation of not the formation of, but the conversion of another type of energy. Okay, it could be heat. Okay, but whatever that energy is, okay, that energy is said to be is the change in free energy. So to go from here to there requires a change in free energy or available energy, and that energy that's lost, if you like, not lost, um, that gets converted to another form, is the energy that's required to do work. Okay, so in physics we have the term work. Okay, that's the energy that's required to do work. Okay, so entropy dictates that something happens. Okay, so the universe is trying to go from low entropy to high entropy. Okay, it's the reason why the sun burns so intensely. You've got this large ball of energy in a relatively small space. I know the sun is massive, but in terms of the solar system, in terms of the universe, it's very small. um, and so an entropy doesn't like the sun being uh, all condensed up with high energy. It wants to go to high entropy, low energy. Okay, and that's why the sun is burning, releasing um, some of the energy into heat energy and other forms of energy. Okay, now for a reaction to happen spontaneously, this free energy, there has to be this difference in free energy. Okay, so if you look at this bar here, this is the reactants. Lots of energy, lots of free energy or available energy in this, and look at the free energy or available energy in the products. It's much lower. Okay, so there's this change in free energy here. Okay, so it's lost some of the energy. That energy has been converted to some other form. Maybe it's heat. Okay, now for the if the reaction is favorable, we need this situation here. Okay, so the products have lower free energy than the reactants. Then it becomes favorable, then it becomes spontaneous. Okay, you still have to give it, maybe you still have to give it a little bit of a push. Okay, but once you've given that initial push, then you get the reaction. Okay, now things like energetically favorable reactions are because you need to put a lot of energy into the system, okay, to actually produce the product. Okay, so you could think of it as uh, the breaking down of glucose here, the breaking down of glucose here to form carbon dioxide, and or the formation of glucose from carbon dioxide. Okay, we know these reactions all happen. Okay, this is photosynthesis, this is cellular respiration, perhaps. Okay, the breakdown of glucose, the formation of glucose. Okay, one is energy favorable, so it's spontaneous, and the other one is energetically unfavorable, so it's not spontaneous. Okay. Now, uh, in textbooks and all kinds of other reading material that you might come across, you'll come across this delta G, this Gibbs free energy, delta G. Now, if the energy is negative, if it's if you see that the delta G energy is negative, that means it's a spontaneous reaction. Okay, if the Gibbs free energy or the delta G is a positive number, that means it's a reaction which requires energy to be put in. So it's a non-spontaneous reaction. Okay, so this is what we call a catabolic reaction, and this is what we call an anabolic reaction. Okay, catabolic to break things down, anabolic to build things up. Okay, and our bodies are building things up all the time. We need new proteins. We make proteins from amino acids. We make, oops, there's it. Um, we make proteins from amino acids. We make glycogen from sugar. So we're constantly making things. Okay, so our bodies are constantly under anabolic conditions. Okay, but also we need both of these things to happen in our bodies. We need to break down. We need the catabolic processes to release the energy to give the anabolic processes to build things up. Okay. Okay. So enzymes speed up reactions, and spontaneous reactions. Okay, react to form products. So look at, if you look at this, A and B are bigger, which demonstrates that, or which illustrates there's much more of A and B in terms of quantity and concentration than the product C and D. Okay. Now the reactions go in both ways, but because you have lots of A and B, the forward reaction is much faster to produce C and D. Okay. Up to a point, once there's equal concentrations of A and B and C and D, and then the forward reaction is in equilibrium with the reverse reaction. So both these processes happen at the same speed. Okay. Now you can think of this as in the absence of enzymes. Okay, this is what would happen in the absence of enzymes. In the presence of enzymes, uh, you can also speed up reactions. Okay, in things like carbonic anhydrase, this will still occur. Okay, because it can produce carbonic anhydrase, and the carbonic anhydrase can convert CO2 plus water to form carbonic acid. Carbonic anhydrase can help to dissociate carbonic acid to form carbon dioxide and water again. Okay, but there are other enzymes which only mediate forward reactions and not so much the reverse reactions. Okay, so it depends. It's different from enzyme to enzyme. Okay, so the central principle is that enzymes speed up spontaneous reactions. Okay, enzymes speed up also non-spontaneous reactions as well. Okay, so anabolic enzymes which are required to build things up are anabolic. Okay, so enzymes also speed up non-spontaneous reactions as well. Okay, so another feature about enzymes is that enzymes speed up spontaneous reactions, but okay, the enzymes do not determine the equilibrium. Okay, so if I go back, okay, there's an equilibrium here. Enzymes don't determine the equilibrium. Okay, this dotted line here represents the equilibrium here. Okay, and this dotted line represents where the forward and the reverse reactions are happening equally. Okay, so enzymes don't determine the equilibrium, so that's a central feature about enzymes, so please do remember that. Okay, but what they do determine is how quickly they reach the equilibrium. Okay, in other words, they speed up reactions. Okay, so if this blue line represents the reaction which reaches equilibrium. Okay, but look how long it takes. So this is time here. Look how long it takes. Okay, and this is the red line demonstrates in the presence of enzyme, and look how quickly it reaches the equilibrium. Okay, both of them reach the same equilibrium, but the enzyme gets there much quicker. Now let's look at how enzymes actually speed up reactions. So let's look at the mechanism by how this happens. Okay, so like we said, these reactions happen natural. These reactions happen naturally, whether there's enzyme present or not. But enzymes just speed reactions up. So how do they go about speeding things up then? Okay. Now, if we look at this statement here, the reactants could be quite stable. So let's say these are the reactants. Now they're quite stable. Okay, but the products that are produced are even more stable than the reactants. Again, this has low entropy and this has high entropy, so the reaction will happen regardless. Okay, now what enzymes do is they introduce this transition state here. Okay, they introduce a transition state for these reactants to produce these products. You have to put a little bit of energy in, what we call the activation energy, to produce the products. Okay, that could be heat, pressure, whatever you want. Okay, what enzymes do is that they lower they lower the activation energy. Okay, and how they do this is by accommodating the reactants in a in a form that helps the reaction to proceed much quicker. Okay, now let me try to explain that. Okay, if you want to break A and B up, okay, it's like bending a stick. If you bend a stick and you bend it, if you bend the stick far enough, eventually you'll break it. But in order to bend it that far, you need to put energy in. The more energy you put in, the more energy you put in, eventually the stick would break. And it's analogous to this reaction here. Okay, this bond, you have to put energy into the bond in order to release the electrons. Okay, so you need to, we need to distort the bond. Okay, so, and in order to distort the bond, you need to put energy in. Okay, what enzymes do is they actually form the catalytic site forms a structure which accommodates the reactants in its distorted state. Okay, so in other words, the enzyme doesn't have to actually distort it physically. It just likes the reactants to fit in its distorted state. Okay, um, I mean, I could illustrate that better face to face, but if you can just picture that. Okay, if you picture a bent stick, if you picture a stick which is, well, initially straight, the straight form of the molecule fits into the active site, but what the enzyme prefers is the molecule with the bond in its distorted state. Okay, it favors that position, and so because it favors that position, that molecule, it's that bond gets distorted. Okay, and as it gets distorted, um, it's easy to break up then, and that's what the enzymes do. Okay, the the amount of energy you need to put in to distort the bond. Okay, the enzymes accommodate that by by making that distortion much easier, and so therefore you don't need that much activation energy. Okay, so holding the molecule in its transition state in the absence of an enzyme is quite difficult. Okay, because it requires energy, but with an enzyme, holding the transition state is much easier because the transition state of the molecule fits much better, okay, into the active site of the enzyme. Okay, hopefully this diagram illustrates what I was talking about a bit better. So here's the active side of the enzyme, here's the bond, if you like. Okay, represented as a straight line. Now the bond is held into the active site rather, if you could think of it like I was talking before, a bit tentatively. Okay, so it binds weakly or tentatively. Now the enzyme prefers the bond to be in this distorted state. Okay, so it doesn't have to do anything because it prefers the bond to be in this distorted state. The bond actually forms in the distorted state then. Okay, and once it forms the bond in its distorted state, it finds it, then the bond then breaks. Okay, and those high energy electrons are released. Okay, now this bond here, this substrate here, binds tentatively. Okay, but when it forms into this distorted state, the substrate actually has better binding affinity for the active site. Okay, and so it, because it binds much more tighter, it's held in that position much better, and that bond can be broken then. Okay, now once it's broken, okay, there's no affinity for the enzyme's catalytic site to hold on to these products, so the products don't have any affinity for the active site, and therefore they fall out. Okay, then the enzyme is free to go around. for another round of catalysis. Okay, so this is how enzymes speed up reactions. Okay, now this is a graph just to show. Here's a graph to demonstrate or to illustrate this concept of free energy, or the relationship between free energy and the transition state. Okay, so here's the reactants. Okay, now here's the free energy of the reactants, and here's the free energy of the products. So this is a spontaneous reaction. Okay, low entropy, high entropy. This reaction will happen. But in order for that reaction to happen, you need to put some energy in, demonstrated by this, what we call activation energy. Okay, um, so this is in the absence of enzymes. Look at the activation energy; it's much bigger than in the presence of an enzyme. Okay, so in the presence of an enzyme, the activation energy is massively reduced. Okay, and this is the main mechanism by which enzymes speed up reactions, by lowering the transitions, by lowering the activation energy. Okay, and this is also this peak here also demonstrates the substrate in its transition state. Okay, what doesn't change? So the activation energy changes. Okay, so it's massively reduced, but the free energy of the substrate and the reactant on the reactants and the products isn't changed. So the delta G is still the same. It's just the activation energy changes. Okay, so so the enzymes again don't change the delta G, don't change the free energy, but it does lower the activation energy. Okay, so again, this is a bit of repetition. Okay, so I'll just skip this bit here. Okay. So that's it basically. There's a few questions for you to go through. Okay, which I'll just skip through. Okay, that's it. So hopefully we've been through these learning outcomes now. Okay, we've talked about the properties of enzymes, the role and the properties of enzymes. We've looked at the classification of enzymes based on their based on their types of enzyme catalysis. Okay, we've looked at enzyme specificity and looked in a bit of detail about how enzymes work in terms of their mechanisms, looking at activation energy and free energy and entropy. Okay, so that's it. The next lecture will be on about we'll be looking at enzyme catalysis, enzyme kinetics to be more specific, and inhibitors of enzymes. Okay, so until then, okay, goodbye. Stay safe.